Yeah,
Yes, sir.
object
Yes,
definition
sir.
in
You can.
primate. Okay, so this is an experimental study by Keiji Tanaka in Japan. So these guys have studied the inferotemporal region in monkeys. In monkeys, it's not called inferotemporal; it's called something else, but it's homologous region where the monkey represents complex visual objects. So they've done extensive study of this part of the brain and found out how neurons respond to complex objects. So then I'll talk about a little bit of that experimental work and then show you how some of those results can be modeled. So we know about dorsal and ventral pathways, right? And so this is just a simple standard picture of the brain. You can see the visual system, auditory system, and all that. Now, so inferotemporal area is into object recognition, as we have already seen. So now the corresponding area in monkeys is called TE, right? And it is monkeys inferotemporal cortex. Right here, neurons respond to complex patterns. And what is interesting is that these responses are recognizing the objects, right, with certain invariance to other irrelevant features. So, for example, an object is the same whether the lighting, surrounding lighting is strong or weak, right? Similarly, the object is same whether it is moved around a little bit or whether it's turned a little bit, whether it change the pose and all that. So, when we talk about an object, we are talking about a concept which survives certain manipulations, certain distortions, right, of that stimulus. So, how does the brain represent objects, right, which survive all these distortions? So, let us look at that. Now, what do the neurons respond to when you say a neuron responds to an object, right? What does it respond to? So, how do you find out what patterns neurons are tuned to? So what they do is actually they present some patterns, right, and then discover or try to look for which neurons are responding to that pattern, and then you label the neuron after the patterns that they respond to. So these areas show this TE area in monkey's brain, right, and it's just like inferotemporal, but it's called TE. Now here the Keiji Tanaka group asked a very interesting question. Okay, you present an object and you find response of a certain neuron in the T area to that object. Now, how do you know what exactly the neuron is tuned to? Right, it might respond to this, but you cannot you cannot identify the neuron based on the stimulus. Maybe there is some other stimulus it will respond even more, right? Or even if you let us say there is a neuron which responds to a picture of a apple tree, right? Now you cannot say that this is an apple tree neuron. Maybe it will respond also to a rose bush or to a banyan tree, right? So what is it about that visual object which is eliciting a response in this neuron? So what is the essence of that object which is eliciting as a response in that neuron? So how do you find that out? So that's a more tricky question because you can always present some random pattern and produce and observe some response on neuron, but that's not good enough to identify what is it that this neuron really and optimally responds to. So for that, they have done a very interesting study. They have followed what is called as a reduction method. That is, so here in this picture, you see kind of rows of images. Look at each row independently. Look at the first row. You see this kind of a ball with a lot of spikes on it. Now, let us say there is a neuron which responds to this pattern. Now we want to ask the question: what is it about this object that the neuron likes? I mean, it's such a pokey object. Why does the neuron like it? Now, do you need all these details present in the object for the neuron to respond to it? What if I remove some details? Will the neuron still respond to it? What is the essence of that object which makes the neuron respond to it? To ask that question, they progressively reduce this object image and construct simpler and simpler versions of this image. To a point, it becomes such a very simple geometric primitive. and And beyond the point, if you simplify the object too much, you will find that the neuron doesn't respond anymore. So that's not what the neuron is tuned to. Okay, so these are some examples of reduction. So in fact, this image is organized into two columns. They are showing a complex object and a simplified representation. So this pokey ball on the left top is represented as this kind of a star with eight, uh, you know, 8 corners. Okay, and similarly, this cat's leg with black and white stripes is presented as a triangle with black and white stripes. Now, this cube, which has a corner which looks like a Y, right, is represented as a T. This person, right, with a black head and long white shirt and a coat and black pants, is simply presented as a white circle and with a small black circle mounted on top of it, and so on. Okay, so this is how they're constructing simple geometric versions of complex real-world images, just to see if the neuron still responds to it. So let us see how it works. It's a pretty innovative kind of a study. So in this picture, you're seeing the response of the neuron. So they've given it a kind of a screwdriver-like object, and then the neuron responds to it. So they're showing normalized responses, right? I'll explain in a minute what is that normalization. So then they simplified the screwdriver image. So in the top image, the screwdriver has some shading, right? You see kind of a strong lighting in the center and slightly darker shades near the peripheries. So remove that shading and make it like a homogeneous shade, and then they present it to the monkey. Now the same neuron responds with a slightly stronger response. That means the monkey likes this kind of an unshaded version even more than the original shaded version. Okay, so then they simplified the object even further down. So in the second version, the outline is still preserved. I mean, outline looks like a screwdriver outline. So then they make three simplified versions of it. They make the handle of the screwdriver into a rectangle, the first one, into an ellipse, the second one, into a circle, the third one. The third one actually looks like a lollipop. So then they look at the responses. You find that the one which has an elliptical handle shows even higher response, a response of 1 compared to 0.62 and 0.57, which they got before. The other two variations, the one with rectangle, one with circle, actually showed much lesser response. Okay, so then they went further. So let me break up this ellipse further and see what is it about this elliptical screwdriver which attracts the neuron. So they broke it up into the ellipse and the line separately. When they presented these two patterns independently, neuron hardly responded. So that means this particular neuron likes some combination of an ellipse with a little line segment attached to it. And that's the kind of combination of features which gives highest response. So the one is actually the measure of the highest response of the neuron. So that firing rate you take as 1, and therefore appropriately you scale down all the other response values. Okay, so this is how this reduction method works. Now, similarly, they've done it for lots of different objects. So, for example, look at the top left figure. When you present a circle with a small kind of a blip on top, it showed some response of 1. If you showed only circle, response almost disappeared. If you showed only the blip, it disappeared. If you showed the same pattern but flipped over, it's an 180 degrees turn, again, there is hardly any response. So, neuron is responding to this particular shape of a circle with a little blip. And here again, it's so sensitive. If the blip is kind of flattened like this, again, no response. If you have only half of that thing and you remove the lower half of the circle, it still responding with half the response, 0.49. Okay, and so on and so forth. So there is, so they distorted the blip slightly, and again, there's huge. So somehow that particular combination of features is what is extracting a lot of information, a lot of response in these neurons. And they decrease the size of the blip a little bit, again, the response has even gone higher, it's 1.36. Okay, so like that, you can study exactly what the neurons are responding to. Now, another thing, like I said, when you want to recognize objects, the recognizer should show certain invariances. It should be capable of recognizing the object against certain variations, so should show invariance. So one of those invariances is translation invariance. That is, an object, suppose you want to see a letter A, no matter where you present on the screen, it's still the same letter A. So you should show translation invariance. So that people have, the neurons in these monkeys have shown that property. So there is some level of invariance as we move around the stimulus and translate it, but it showed highest response for object present in the center of the receptive fields of that neuron. Okay, which makes sense because neurons are tuned to certain receptive fields. So similarly, the T neuroreceptive fields are longer because as you go higher in the hierarchy, right, receptive fields are longer. We have seen that in the last classes. Um, so that's why they're able to recognize complex objects. Now, another kind of invariance is scale invariance. So if I write a letter, whether I write it big or small, it's the same letter. I should be able to recognize it irrespective of its size or scale. Now, these neurons in monkey T area also show this property, which you can see in this figure. So they show this kind of a star with 8 corners, and at the smallest scale, the response is 0.68. At the second scale, it is 0.94. Third scale is 1. And slightly higher scale, it is 0.83. So it's pretty robust against scale variation, but it seems to be more tuned to certain scale over the others. That's why it's maxing out at this particular scale. Now, so the response remains fairly stable over a size range of about 2 octaves. So 2 times and 2 times, right? So only over that range, you see a response fall of about 43%, so which is a fairly robust response to scale invariance, so response to scale variations. Another invariance is rotation invariance, but it is somewhat weak because so obviously rotation invariance should not be taken too far. Because if I rotate a letter N, it becomes Z. I don't want to be confused between N and Z. And that's also not good because in the real world, right, gravity acts like a symmetry breaker. Because my vertical axis is important, it's sacrosanct because that axis is defined by gravity. So rotation around that, right, is something. So there's I have notion of vertical, right? So which is different from horizontal. So rotation variance should not be taken too far, and therefore in the response of cells also rotation variance is somewhat weak. Okay, so rotation of the critical feature by 90 degrees decrease the response by 50%, right, in most cells. So if you see this picture, so see the original picture is a white circle and a black circle on top of it, where the cell shows highest response of 1. But when you turned it 90 degrees to the right, response almost fell to 0. Turn it upside down, it is 0.22. Turn to the left, it is again become negative. So it is quite tuned to the given direction. It's not very sensitive. It's not very. It doesn't show much invariance to rotations. I'll skip that. Now, just like in your orientation sensitivity, here also now do we expect columnar organization? Are there columns? That means within a column, as you probe the neurons along the depth, right? Do they all respond to the same objects or object shape? The answer is pretty much yes. So, because if you look at the objects to which the neurons are tuned to, in terms of these reduced versions and not the original complicated versions, neurons within a column seem to be sensitive to pretty much the same object class. Then another thing is if you probe the IT area or TE area horizontally, right? Again, just like in the other orientation sensitivity or ocular dominance, nearby neurons in the cortex respond to similar objects. So therefore, look at this. The objects are numbered as 1, 2, 3, 4, right, and 5, 6, 7, 8. The top and the bottom. Now, in the middle figures, it shows the cortical areas over which these objects produce some response. So object 1 produces a response over these areas which are linked to number 1 in the second figure from top. So now object 2 is producing activation in areas that is connected to object 2, with a dotted border. So you see that object 1 and 2 look similar, right? And also even in terms of responses, there's some neurons which respond to both object 1 and 2. So there's an overlap. So and so on and so forth. You see that similar objects, so nearby neurons respond to similar objects. So similarly, even for faces, so you see that they have shown this kind of a bust image of a head, and then they change the orientation or pose of the head. And as you gradually change the pose, so obviously you know that pose has two angles. right? So it has 2 degrees of freedom. So as you change these 2 degrees of freedom, the neurons which respond to each pose, that area also, right, moves gradually, right, in the 2D along the over the 2D surface of the cortex. So each of these regions that you see, these ellipse-like regions, they represent the neurons, the set of all neurons that respond to that particular pose. Okay, now some of these results can be modeled using a very simple CNN model. This work we have done as a dual degree project, with a student called Rohit some years ago. So I'll quickly mention that. So Rohit has taken a CNN and trained it on images of size 50 by 50. So he has taken 12 images and from the same Tanaka set and trained the network. So images he has taken are these. So this picture of the apple and the ball with shading. Sorry. Shit. So the ball with shading and all these things, the cube and so on. So you see that you can see certain responses to different objects. So then he goes into the network and studies if there are single neurons which respond exclusively to certain object classes. Okay, so we found that certain nodes or neurons remain non-responsive to all classes, while some nodes respond to all classes. Okay, so that such neurons are no good and they are not of much use to us, because we need to find out if there are neurons which respond to one class strongly and only that class and nothing else. Then we say that in such neurons are tuned neurons or tuned nodes. And not all neurons have this kind of a property, so we need to find a score to isolate these tuned ones from untuned ones. So first of all, how do you know that a neuron is tuned, right? So for that, we design this kind of a score or a tuning factor. It's called T with the subscript Ki. So what this says, it's a tuning factor of the kth neuron for the ith pattern. So it says how strongly is the kth neuron tuned to the ith pattern. So which means for the ith pattern, it should show a response. For all other patterns, it should show much different response. So if the response is, let us say, 1 and -1, or 1 and 0, the two extremes of the response. If it shows 1 to the ith pattern and 0 to all other patterns, that's good tuning. I have no problem with that. Similarly, if it shows 0 to the ith pattern and 1 to all other patterns, that's also equally good. The neuron must show a kind of a distinct response to a given class or given pattern, as opposed to all other patterns. Okay, so if we do that, then this tuning factor will be a high number. So basically that can be then defined as xi is a response to the ith class or ith pattern, xj is a response to the jth pattern. Difference of this, this is power on this square, and sum this over all patterns, okay, and find the score for the kth pattern. So you look at the response of the kth neuron to all the patterns and calculate the score. So then a large TKI denotes that the response of the kth neuron for ith pattern is different from its response to the rest of the patterns, right? And similarly, so if TKI is small, it means that the neuron responds in about the same way to all patterns. So it does not have much tuning. So we're not interested in such neurons. So now we are interested in neurons which respond uniquely to a single or a few classes. So therefore, we look at neurons which have high TKI values. So here again, neuron might, so basically what TKI says is the response of the neuron high to ith pattern compared to all others. Okay, so now you can calculate similar tuning for other patterns also. So therefore, so you take the TKI value, response of the neuron to ith pattern, divide it by the average of the response to all other, average of the TKI score to all other patterns. Okay, so that number tells you how strong the tuning of the kth neuron is on the whole, right? It doesn't matter to which pattern. So so if we looked at the R values for all the neurons, and we found that the highest value of R is about 2.3, and we just fixed a threshold at 2.0, and they found that 15 neurons have satisfied this cutoff. So, and then we found that the neurons are tuned to, among these 15 neurons, they're tuned to 4 patterns. But if you set the threshold 1.7, we get 42 neurons which are tuned to 8 patterns. So now let us see what happens if you perform the same kind of a reduction method that the experimenters have performed on real monkeys. So for example, on the right side you see neuron responses, left side you see the pattern which is presented to the network. So suppose look at the top left pattern, which is kind of, I don't know, some lollipop kind of thing. 12 neurons, the responses, sorry, for this neuron, this whatever this kth neuron, the response of that neuron to all the 12 patterns looks like this. So for this particular pattern that you are looking on the seeing on the left side, the response is negative, like minus 0.6 or something like that. But for all other patterns, the response is positive. So it shows a distinct preferentiality to this particular pattern. So now, just like in the experiment, we try to follow the reduced versions, present reduced versions of the image, and then see what happens to the response. And these reduced versions also, we didn't make them up. We just use the same things that were used in the experimental study, and see if the neurons in the model also behave in the same way. So in this first version of the reduction, we remove that stick which is stuck to that lollipop, and presented the remaining thing. And we see that the response hasn't changed much. It's still about 0.6 minus 0.6, just like before. Then we made this in the original thing. This lollipop slightly more elliptical, slightly more regular in shape. When we did that again, the response went down a little. It's going towards 0, right? Slightly. That is, it's now minus 0.5 instead of minus 0.6. Then we made it like perfect ellipse, right? Even there's little dots and there's noise, the speckles, all that is removed, right? And so then the response has gone down drastically. Which means there is something about this particular shape with all the noise, with all this, this little specks on it. All that seems to be important for the neuron. That when you remove all that and present a clean image, its response has gone down. So then we took the darkened version of it. You fill the circle with the ellipse with black color, and now the neuron is looking like just like the response to other classes. You can't distinguish. So it's lost its tuning. It's not tuned to this kind of an image anymore. Okay, so similarly we have done it for other images also. Again, hand and this kind of a picture with 3 pointed regions, and cat is reduced to a simple kind of a simple outline filled with black color. So again and again, you see the same kind of trend. So the point of this exercise is even such an involved experiment, right? What was done with cats' object recognition, properties, can be simulated using a simple CNN, right? Provided you probe the neurons in the right way, right? And just like what they have observed by presenting the animal with progressively reduced versions of the input stimulus, right? We have also observed the same kind of response of neurons when you present it progressively reduced versions of the input stimulus. So thing is, so you don't need a very complicated biophysical neuron model, right, to simulate complex neural response of the real brain. Okay, so let us do one more model like this. So this model was proposed by Risenhuber and Poggio, published in Nature Neuroscience in 1999. So they present a hierarchical model of object recognition, again in the inferotemporal cortex. So like I just mentioned, right, not only Tanaka's group, but many groups have shown that cells in the inferotemporal cortex show this kind of a scale and translation invariance. So the question that these people are addressing in this model is, how is it possible, and how do they acquire this kind of a property? So in this figure, they are citing some experimental study. So by, I think, Logotethis. So where the experimenters present paperclip objects. Okay, so these are wireframe objects in 3D, These are presented. These are of course simulated and presented to the animal. So now they present certain distortions of it. So they vary the scale, they move the object around, let us show translation, and rotate the object on the vertical axis, and so on and so forth. And they are looking at the response of the some inferotemporal neuron area. So when you rotate the object on the y-axis, it shows that there is a sharp tuning, because a rotated object is in the vertical axis is not the same object. It can be very different objects. So you don't want too much of invariance for that. And just like in Tanaka's experiment, in Logoteth's experiment also, neurons are not very invariant to rotations. Okay, but and the response to the 10 best distractors, that is, the neuron is responsive to some pattern, some particular paperclip pattern. But if you show other paperclip patterns which it's not tuned to, it shows very low response, that you can see in figure B. But in visual angle, that is scale, basically if you move the object far, right, the visual angle that it subtends on the eye, it goes down. So that is, that is like scale variation. So the neuron is fairly scale invariant, as you can see in figure C. Right. Similarly, as you translate the object around, again here also that is somewhat in a translation variant. But in case of so in figure D, in the top figure, you can see where the object is presented. In the first one, it is presented in the center. Second one is presented to the right top. In third one, it is presented to the right bottom, and so on. So the first three, it is showing high response, but 4 and 5, it is showing slightly lesser response. So the question is, okay, you can easily get object recognition, but how do you get this invariance, right? And how do you achieve this combination of invariance and specificity? So invariance means non-specific. Specificity is opposite, right? So how do you achieve this combination? So the answer to that is by designing the right kind of hierarchy. And that's not surprising, because you have noticed that the inferotemporal area, or TE area, is at a very high level in the hierarchy, high position in the hierarchy, because there's so many layers of neurons which come before that, starting from retina, where already you have three layers, and then you have 6 layers of LGN, and then V1 and V2. And so at the end of a huge hierarchy, the visual hierarchy, you have inferotemporal area. So you have enough hierarchy sitting before it in the visual system. So how do you construct this hierarchy? So they have taken data from visual system, experimental vision science, and constructed this network. So for example, we know that there are these simple cells which respond whenever there is a bar located inside the receptor field, especially in the center of the receptor field. And then there are complex cells which respond whenever there's a bar located anywhere inside the receptor field, as long as the bar has a given orientation. So they take a bunch of these simple cells and then combine them into a complex cell, so that the response of the complex cell is somewhat translation invariant with respect to that bar. Okay, so you take these simple cells at one layer and project them to the higher level. So then that means you take multiple simple cells at a lower layer, right, and then project them to a higher level. So see that the simple cells that you use to combine here all have the same orientation. So in this illustration, you take 3 simple cells, all of them responding to the horizontal line, and project them to one complex cell, right, and then use some kind of a OR gate kind of relationship. That means make sure that the threshold is somewhat low for this complex cell. Then it will respond whenever any of them is true. That means, right, so that will give you some kind of a translation variance because of this kind of arrangement. So like that, they have multiple levels. They have a simple cell layer in the bottom, and then so basically they put receptive fields all over. They cover the entire input image using receptive fields. Now for each receptive field, a given simple cell responds whenever there is a bar of a given orientation in the center of a receptive field. Like that, they cover the entire input image, and then these cells are combined, like we just described, to construct one layer of complex cells, C, that's called C1. Then the complex cells are again combined to produce composite feature cells called S2. So these are combinations of two bars, right? So forming angles. And if you remember, I mentioned that the secondary visual cortical area, so that is V2, right, it responds to this kind of angle-like features. So the S2 in this model is kind of like V2 in the real visual system. Then they combine these S2 also in just like in they construct complex cells from simple cells. They construct complex composite cells C2 by combining the S2s in such a way that you get some level of translation variance. So that's it. After that, they just project it onto one layer of what is called view tuned cells. So these neurons respond whenever you show a paperclip object, right, from a certain view. And that last layer of weights are trained using simple delta rule, or like it's like your perceptron. So you train that. So it's a very simple model. And so it's trained on 80 images, 20 targets, and 60 distractors. These are 64 by 64 size images. The images are images of paperclip objects, like those shown in the figure here. So we want to understand position variance. So as you move the object around inside the input image, how much does the response vary? And scale invariance. As you show the object at different sizes, how much does the response vary? Right, and rotation variance. As you rotate the object, right, around some axis, how much does the response vary? So you see in this figure that figure A shows the response of a given neuron, which is tuned to that object, right, as a function of the scale. And you see that when the scale is too small, it doesn't recognize, therefore the response is too small, right? But as you increase the scale beyond a threshold, you see that the response is fairly constant. So it is quite scale invariant. From about a similar size of about 30, right, to 160, the response is fairly flat. But the viewing angle, the response of the neuron to the viewing angle is not quite stable, right? Because from different angles, the object might look different, right? So between about 70 degrees to 110, so 100 degrees, right? So from 90 degrees to about 100 degrees, already the response has fallen by about 50%. But it is quite, shows very strong translation variance. Because as you can see that the network is designed to be translation variant. Because when you go from simple cell to complex cell, you are encoding some level of translation variance in that design. So you see in figure C, you can see translation variance. Okay, so one more question they are asking is the neuron responding to certain pattern in the input image, or is it just responding to a bunch of features? So that means if it's just responding to features, it should respond to them no matter where the features are, right? So for example, just to give a small illustration, right? So you can, yeah, so let us say I'm responding to, okay, respond to letter A. Now, am I responding to this entire pattern? Am I responding to certain features? Because the A has an angle feature, a T junction here, another T junction here, right, and two line terminals. So am I responding to a certain complex of all these features arranged and connected in certain specific fashion, or am I responding to them as independent features? So if I give this and this and this and just two lines like this, located anywhere and present them as an image, will the neuron respond? Okay, so the answer to that is in this result here. So they have taken an image and then cut it up into small pieces and jumbled them. Okay, so that number of pieces into which they cut the image is given is this number of bits, number of blocks in it. So So when it is 1, when that means you're presenting the whole image and response is 1. When they made it into 4 pieces, the response already fall into 0.6. 16 pieces is respond to 0.2. That means they have the same pieces in the image, but they're all jumbled, which means and the response is not the same. It's really falling exponentially with the number of pieces. So which means that it is respond to that and such that pattern of all those features and not to individual features, as if they are all independent features. Okay, so what we learn from this also again is you don't need a very complicated, bio-inspired model, right, to produce some of the responses that you see in real brains, right? And so that's what these people have shown. So here, very easily, they were able to construct a network which shows scale invariance and some level of translation variance, right? Even the kind of view invariance that the network was shown is similar to the kind of view invariance that neurons of a real monkey brains are showing. Okay, so this is very important, I think, because this course is all about one of the key ideas that we want to show in this course is how very sophisticated observations from real brains can be reproduced using quite simple models, right? And both of them are decent illustrations of that principle. So next class, I'll show you how deep networks are being used to explain a lot of very detailed neurobiological data from not only visual system but also auditory system, right? And after that, we'll move on to another topic. So thanks a lot and happy Diwali, everybody.